Thank you very much. I would like to start, thanking the foundation. Really, it's a great privilege for us. It's a real privilege for us to be here. Thank you for inviting us. Thank you for, taking us in account for this presentation. And I'm very excited, and thank you, all of you, for being here. I am a clinical neurologist, and I have been working in neurology for a long time. I have been in the industry trying to develop medications for the last 22 years, and I have been in multiple areas, different areas. And I decided to join Xenon recently as a chief medical officer, just around 6 months ago. And this program, these topics, trying to help in the way that we're trying to do now, is what motivated me to make this big step. I have been working all my life in large industry in general. I work for Merck, I work for Teva, I work for big companies. But having the opportunity to come and see what we can do to help your kids and to help people that has that difficulties in life is what motivates me and makes me awake every day. So it's a great privilege. It's my honor to be here. These are my forward-looking statements. Let me tell you about Xenon, and it's just one slide. Xenon is a pharmaceutical company based in Vancouver, Canada, small company with great expertise in genetics and very interested in general in channelopathies. And that's where we're putting all our efforts now, in general, in channelopathies. And of course, when you work in genetic and channelopathies, epilepsy is one of the main areas to be working on. So that's where we dedicate our time. We have a very strong discovery group, and we have now a very strong development group. So we are fully committed to discover and develop new options. I'm going to talk a little bit about the KCNQ2 encephalopathy, but of course it's going to be very basic. And you have heard from Dr. Cooper, and you will hear from others, a lot more science about this. But it's very simple, very simplistic way that I'm going to show you, so I can go faster to what we are doing, trying to improve, to try to give some value with what we are doing in every day. The nervous system is, and it's very complex. Biology is simple, it's very simple, but at the same time it's very complex. And in general, you know that we have the central nervous system, that is, basically the brain and the spinal cord. And we have the peripheral nervous system, all the nerves that conduct all the signals in the body. And in general, the information has to come from the brain down to the nerves. For example, for all the motor system, to move, we have to send the message from the brain to the muscles. And that message have to travel, through different parts of the brain, of the nervous system to be able to move a muscle. But on the other hand, information has to come from the periphery up to the brain for our sensory system. So if we want to feel something, the information goes from our foot to the head. And in the head says, oh yes, I'm feeling hot, or yes, I'm going to burn here, or yes, my right knee is in this or that position. So information has to come up and down in the nervous system, coming from the brain to the periphery, but also coming from the periphery to the brain. And at the same time, the information has to circle in the brain. There's a lot of connections, a lot of things that have to happen to make things happen, to make things move, to make us feel something. So we all know that we can feel something without any external stimulus. We all know that we can move a leg or move an arm without wanting to do it. And it's just because the connections in the brain may trick us, and they may just start sending signals up and down without control. So the message has to, go up and down or circle in the brain in different ways. But the neurons are those cells that transmit that information. They have to take the information from one side to the other. And it's just beautiful what happens, because that information travels just with electrical activity and the exchange of potassium and sodium in and out the neuron, starting in a specific part of the axon, is what makes the information travel from one neuron to the other. So changing potassium, changing sodium from the in and out, is what makes the information move. So one neuron sends the information to another neuron, just changing that. So of course, if we have a problem in the exchange of potassium or sodium, something has to happen. And the information then is not well transmitted, or is too much, or is too low. So the way that that information goes and travels in the neurons, well, can be measured, can be observable, if you see the electrical activity. So if you see the electrical activity, you see, oh, the potassium is coming out, oh, the potassium is coming in, oh, the sodium is coming out, oh, the sodium is coming in. So the easier way to see it is just to see what is the electrical activity. But when the information has to go from neurons to neurons, oh, it gets very complicated. There are so many things that happen. So many proteins that go there, so many neurotransmitters that affect that have an impact on how the information is transmitted, that gets things very complicated. So as Dr. Cooper said before, for example, changes in proteins or changes in genes, I am absolutely convinced that there is no one change will not have the same effect in two people. It's very complicated, it's very complex, and all of us are still individuals that respond differently. Now, let me give you one word about epilepsy in general, and I will make a very important point in how is our interest in epilepsy and how is our interest in encephalopathy, because it's different. But epilepsy, of course, is this disease, this chronic disease where seizures is the main landmark. And seizures may be, we are interested in where the seizures are beginning in the brain. What is the level of awareness of a person of that seizure? Because you may have a seizure and you be aware of that, or you may have a seizure and you may not be aware of it. Or other aspects of the seizure. Is my seizure a movement? Is my seizure not a movement? What is the problem? So these three things are what make the classification of seizures now, a little bit complex, but important for us. So when you go and you want to characterize what type of seizures a person have, in general, they're going to ask, okay, the seizures start in one part of the brain, or the seizures just start, suddenly in the whole brain, or, definitely we do not know what is happening. And each of them have, different levels of if the person is aware or if it's not aware, if it is a motor, if it is a no-motor seizure. So that's what basically is the classification of seizures today. So let me go back a little bit to this firing. So we know that in KCNQ2, or when the potassium is altered, it has a problem in the communication in the neuron. These neurons start firing a lot, and this is just the electrical view of what could be happening in a very general, simple way. So when this firing happens and you open the potassium channels, you just try to restore and calm the neuron. And that's the principle in, when you have KCNQ2 and you have this difficulty in the potassium channels, and you bring a medication or you bring anything that can help open those channels and enhance the activity of those channels, then you may facilitate the exchange of potassium, and you may bring these neurons back to where, the state that they should be. So in some way, as Dr. Cooper showed, this is the spectrum, I would say, of issues of what is happening in the clinic. But, you can, if you are able to open that, you may be able to, increase the potassium exchange, and we expect that there is an impact then in the phenotype and what is happening with the patient. So that will affect not just the seizures, but also may have an impact on the development, or may have an impact in general in what is in the condition. So this is KCNQ2, and I'm not going to go deep in detail. This has been you are more, to teach me about this disease. And of course, the experts that are in the room, they know a lot more than me about, specifics of the disease. But this is a traumatic condition, and we need to do everything we can to try to help and try to solve and try to make improvements in the identification, but also in the treatment, improving the quality of life of these patients. So what about ezogabine? Let me tell you a little bit about what we are doing with ezogabine and how do we see the problem with ezogabine. In this table, I'm going to show you that, you're going to see here a list of anti-epileptic drugs. and the different channels or the different mechanisms of how these antiepileptic drugs work. So as you can see, ezogabine is very specific. It works in potassium. And other medications, a lot of other medications may touch the multiple different channels and multiple different proteins and mechanism of action. But ezogabine is relatively specific, very specific to potassium. So that makes ezogabine an interesting drug to treat or to add value to patients with KCNQ2. And it has been proven. Retigabine has been proven to be efficacious in focal seizures multiple times. So you can see, this is the results of basically of the phase 3 studies, when you see how ezogabine at different doses is better than placebo. It's better than placebo in the 50% patients that respond more than 50%, patients that respond more than 75%, or even in patients that have total control of the seizures. These studies were done some time ago. But with those studies is how Glaxo get the approval of the medication for low focal seizures. And then ezogabine, as a unique potassium open channel opener, was in the market. So it's proven, it's a drug that has been proven to be efficacious in focal seizures in adults. And then there came a lot of reports that could show about how this drug may benefit patients with KCNQ2. But this is when things get a little bit difficult for us in the industry, or for us that are developing medications. That is that it seems that the physiology is there, pathophysiology is there, the mechanism of action is there, the reports are there, but we need to prove that this is real. We need to prove that this is the way that we can go back to tell the agency, we can this should be indicated for this patient population. When a medication is in the market for other indications and a lot of off-label use starts happening, okay, that is something that is very difficult to control and nobody can control, but the drug is available for different uses. And doctors decided if they use it offline or not, off-label or not. And that was the case with ezogabine. That ezogabine was in the market, and multiple reports start appearing about ezogabine and benefit in KCNQ2. It was not a full black and white response, say, every patient that is in ezogabine responds 100% or not, etc. But definitely, the drug was start being used in these kids, and some benefit was seen in some of them. But as I said, the idea is now, how can we prove that that happens? Probably there was not much interest in demonstrate that because the drug was in the market. But the problem is that ezogabine is not in the market anymore. Ezogabine has some interesting characteristics in the adult patient population that was studied. Some of you, maybe had your kids in ezogabine when it was available. And ezogabine forms these dimers. As part of the metabolism, it forms these dimers in the chemical constitution of the molecule. And those dimers are responsible that the molecule gets blue. And that causes, when in the past, when people try to, macerate the pill or try to play with it, it gets blue very rapidly. And it was then reported that some patients have the blue syndrome. They get blue in the skin, areas of blue in the skin or in the mucosas. And even there was reports of getting blue in the retina, blue spots in the retina. And at that time is when the FDA led a warning, with a warning on the medication because of these side effects. There was never, proven that there was an effect on the visual acuity or in the visual problem. No, it was not proven that, but it was not also discharged if that could happen. So there was a warning from the FDA on that regard. In addition to that, ezogabine caused urinary retention. And because of the urinary retention, it was manageable. Some patients manage in different ways. But this adverse events profile and with what was happening at that time let's say in the field of epilepsy, Glaxo decided to took out it from the market. Then Xenon, of course, working in different areas in channelopathies, and because what we are doing, and I will show you a little bit about some of the molecules that we're working on, we think that we have an opportunity here to help. And then is when we said, let us think if we can do something with ezogabine. Ezogabine is not available in the market today, but we start exploring what can we do to bring it to the market again, specifically for KCNQ2. And one of the reasons why we're thinking like that is because it's the fastest way to bring some value. It's the fastest. We are working in other programs as I'm saying, one of them I'm going to show it to you, but this probably is the fastest way that we can. So there's this act in the FDA that is the Orphan Drug Act, probably some of you know, where you can apply for an orphan disease designation in a medication. And if the agency provides you that designation, let's say, you have a significant benefit to interact with the agency. You have a significant benefit in the approval process, let's say. We applied for that, and we got orphan disease designation in KCNQ2 for ezogabine in June. And I think this was a very good step for us that saying, okay, we are very interested. We want to make this happen. So we have today orphan disease designation for ezogabine in KCNQ2. And as you heard earlier, we decided, because of what we are doing, and I'm going to show you, that we are full speed in the development of ezogabine, trying to bring it back for KCNQ2 patients. We announced publicly that we are committed to this. We are making significant progress in developing our pediatric formulation. And this is, I want to call your attention, this is probably one of the most difficult things that we are doing now, more challenging, difficult, complex. In the past, of course, ezogabine was not developed and produced for pediatric population. And the dosing, the taste, the way that it binds to the bottles, the way that it binds to plastic, all these things have to be taken account. And we need to develop a formulation that we can dose and we can give the appropriate dose to our kids. So we are, doing all our efforts to develop that pediatric formulation, and we are making progress. But it's still very challenging. And that's a niche that we have to overcome. We have, we are working with the experts, with the people that are here, trying to, help us in define what is the best way to develop it, what is the best way to bring it back to the market. But let me give a little bit of information on what is the complexity here. To develop a drug, as I said before, it is not just, I have this medication, I'm going to try it in 20 patients, it seems that they're getting better, I go and say, please allow me to sell this drug, and it's done. It's not like that. It's very difficult to get that because we need the balance of understanding what is the benefit and understanding what is the risk in the specific population. So the development of a drug implies a lot of discovery efforts and a lot of preclinical efforts, and it is a very long process. And later, then you have the phases of the studies that you have to conduct. You know probably well this, but at the beginning, you have to do what we call phase 1 studies. That is small studies, usually done in healthy volunteers, and you give the drug to a healthy volunteer, and then you see what happened. What are the adverse events? What are the complications? And if that goes well, then you go to the phase 2, when you say, okay, let me see if this drug works. What is the dose that I have to use? And I try different doses. I try different regimens. And if I said, yes, it seems the drug works, then I have to go to the phase 3, large studies. Usually in indications, large studies, when you say, okay, or what we call confirmatory trials. That is when you go and you test the drug, usually against placebo or usually against other drug, and you see if there is a statistical significant difference, and you can convince the agencies and everybody, this is real. The effect of the drug is real. And once the effect of the drug is real and is confirmed, we then submit to the agency for approval. And the agency says, yes, you can go and offer this drug for patients. And that happens after you have all this risk-benefit balance clear for patients. So it's complex. It's very difficult. But in this case, when we evaluate this case is different. Because preclinical, clinical, phase 1, phase 2, phase 3, all has been done for patients. They are adult patients. Yes, they are adult patients. But all has been done. And the great effort that has been done in the past, trying the medication and with case reports, and the families that decided to put their kids in ezogabine in the past, give significant, very important, valuable information that we can go back to the agency and tell them, hey, we have something here that we don't need to do everything. It seems that we don't need to do everything. Probably we have an alternative. So with that, we approached the agency. And I'm going to tell you a little bit about the importance of these studies. When you do these studies, This is a very important slide for me. When you do these studies, specifically in phase 3 trials, you want to confirm if the drug is safe or not. The drug may be very safe in an adult, but not in a kid. Maybe very safe in a kid, but not in an adult. We do not know. We have to study that. We need to define very well the risk-benefit. And to do that, we have to put in place studies that have clear opportunity to respond to the question if it is real true what we are seeing or not. So we need to have inclusion criteria to say, we need this type of patients or this type of events to evaluate so we can really compare and say, yes, the effect is real. And then we can have the drug approved for everybody. But in any study, it's like that. In any indication, it's like that. Actually, to be honest, sometimes we are criticized that we study populations that are not real populations. They say, you just select the patients and just select the things that you want to see, and then you go out for a lot of people. That is true. In a lot of diseases, that is true. I do a study, my previous experience, for example, I did a study in migraine. I studied 1,800 patients. Drug was approved yesterday. Now it's going to be used in millions. What is going to happen? I started it in 2000. But now millions. Let's see what happens. We have to now be vigilant to that. But it is very important to have these criteria because otherwise the noise is not going to allow us to see the effect. And the objective here is to be absolutely sure that if we go and we said, yes, this drug works, we have cleared the profile of the drug. So everybody knows what are they facing. So this is challenging, very difficult. The inclusion, the exclusion, how do we select the patients to be able to prove that what we are thinking and that we are saying is correct. And the other thing very challenging is this, is what we call the endpoint. What are we going to measure? Because, for example, in KCNQ2, it's very important. The seizures are an important feature, but it's not the only feature. There are other things. Development is probably more important for a lot of you. And for us, of course, it's very important. So what are we going to measure to tell the agency, we have here a clear difference. Please allow us to go out and offer the medication for patients. So the endpoint, what do we measure? We have to agree with the agency. And sometimes the agencies have clear opinion on that. They said, no, this is what you have to measure, depending on the indications. So in pain, this is what you have to measure. You have to measure pain like this, like that. So they have a pre-specified thinking in what do we have to measure. So we have been in touch with the agency for some time, and we have very good interaction with them, and we have been making significant progress. We proposed to the agency, first of all, how do you see if we go with ezogabine in pediatric development? We want to develop a drug for pediatric population. We want to put in place a safety assessment for these kids. Will you agree with that? Or maybe the agency says, no, with what you have in mind, we don't want that. This drug has some things that we don't want back. But the agency says, yes, we agree. They're responding in writing to us that they agree with us moving forward with this program. We asked the agency if one study will be enough. Usually you need 2 studies. In general, to get a drug approved, you need 2 studies, 2 phase 3 studies proven to be the drug is efficacious to be able to have the drug approved. So we asked the agency, will be just 1 study enough? The agency says yes. We can review that with you, but they agree. We told them it's difficult to do a placebo-controlled trial. We cannot say, you're in placebo, you're inactive, let's see what happened after 6 months after, et cetera. But we can play with that. We can have a crossover trial where, you're one time inactive, one time in placebo, et cetera. And is that design acceptable for the agency? Will you be willing to work with us in a design in that model? It is, that type of design usually allow us to have a better statistical opportunity. But we didn't know if the agency will agree with that, if they would like us to agree with us with moving in that direction. They said yes. They said that's possible. We can go with that. But the safety was the other issue. We asked the agency, for example, if we enroll babies in this study, it's going to be difficult to monitor the urinary retention. It's going to be difficult to measure different things in these babies. Will you agree if we And we propose some specifics in monitoring for urinary retention, some specifics in how we can monitor, the eye, for example, the color of the eye, etc. Will you agree with that setting of things that we are proposing to do this monitor? And they said, yes, we agree with that. So we have made incredible progress. We think that the agency and us, we are receiving very good feedback from them. We think that we're in a good page now to move forward with a proposed study to them, and that's our commitment. So in general, our study today is, we are still defining what is the best way to do the study. We are still defining how can we do it and where can we do it. We need a lot of help if we move forward, and we can move forward with this. But in general, the agency is asking us that the way to measure in a more objective way is seizures. We have to prove that we have an effect in seizures. So because of that, our endpoint, as I said before, is seizures. We have to measure seizures. So we are putting in place a study that is going to be, having this crossover design probably. And at the end of the study, we will, of course, we cannot just let the babies there without any options. So, of course, we will have an open label extension where everybody will be able to go on continuing the medication. We are exploring how can we do this, how can we offer this. We are exploring and trying to confirm with the agency, and this is an ongoing dialogue that we have with the FDA specifically. We are in permanent communication with them and trying to see how can we optimize this and if there is any different way to do it. But definitely we are receiving very good feedback in what is the next step moving forward. So for us in 496, as we call it, ezogabine, for us in our programs, we need to work with the FDA in the details. We are, as I said, we are sending permanent information with them, and we have communication with the agency. We need to build a network to be sure that we can, execute in the trial. We need to build a network to be sure that we can enroll the patients, that we can offer everything they need for the trial. And we need to finalize our pediatric formulation. We have a lot of work, that we have done a lot to develop the pediatric formulation. We have made significant improvement. But that's a piece that we need. Without that, we will not be able to start a study. So we are working very hard. And as I said, ezogabine it has chemically significant difficulties that we have to overcome. This molecule, for example, sticks to plastic like crazy. If they get close to a bottle, they just stick to the plastic and they just don't move from there. So we need to be sure that the dose that the kids are receiving is the appropriate one. So we need to create, microparticles. We need to play with it in the way that we can then dose properly. We are improving, for example, the color, the taste, being sure that it's compatible with different things so we can offer the right medication for the study. We need to prove this. And again, another important thing for you to know that probably you know is that anything we do in this study has to be very well aligned with what it's going to be having in the commercial later, once it's approved. So we have to use the formulation. We have to use the way that is going to be then available for patients. And that is taking a little bit of work, a lot of work for us. It is very challenging. But we are moving forward. We are putting a lot of energy in this program because we know we are totally convinced it's the fastest way to bring something that can help. We expect that we can start this study probably third quarter next year. I hope that second quarter of next year we can start the study. And we are, of course, exploring all the options. We are exploring options to bring, compassionate use as soon as we start a study to patients that do not qualify for the trial. We are exploring and working with multiple experts in what is the impact of ezogabine in development, how can we measure it. We need, to work very close to see what is the natural history and understand how we're going to be comparing and against what. What do we need to measure in the studies? What do we need to measure in the patients that would like to go in a compassionate use? How do we measure that? To really show the benefit, to really show if there is benefit or not that can help other patients in the future. It's a difficult decision, but an important responsibility that all of us have is to be sure that we do the right studies so we can, have well-informed parents in the future if they decided to go in one treatment or the other. So let me tell you a little bit about SEN-1101, that is the other molecule that we have. You heard about 1101 in a previous one, and 1 year ago when my colleague, Paul Goldberg presented, in this forum. So I want to show a little bit about 1101, that is our new KV7, opener. Where are we with this molecule? This molecule is a novel KCNQ2 modulator. It has the same, let's say, in principle, mechanism of action as ezogabine, but we have significant improvements in the molecule. This molecule has a very much potent in vitro and in vivo, quality. It has a very significant improvement in the PK, so the weight and the duration of the drug in the system is longer. We can dose, less frequent. It's predicted not to have this liability of the dimerization, so do not create these dimers. But is important because do not cause this blue color. And we have important advances already in phase 1. So we already did the first set of studies. We already did the whole set of phase 1 studies in adults, and the drug is looking very, very promising. So this is one way to show how one of these drugs may work, or what is the effect in the biology of the brain. One way is to do what we did, that is transcranial magnetic stimulation. So it's a simple study, I will say, where with what I said, we told you before, is that you take a patient, in this case, you take a human volunteer, this is a healthy person, and you put a magnet in the brain here externally, and you give a stimulus. You give an electrical stimulus. And what happens is that electrical stimulus, of course, activates the cortex, and goes and travels and goes to the finger. For example, if I activate the area where the finger or the hand is moving, if I activate that, then the subject is going to move a finger. Because I activate here, then the finger is going to move. So I can measure what happened from here to the finger, and I can see if there is an effect of any medication or anything in these healthy volunteers. But at the same time, I can do EEG. So I can do the stimulation, but I can measure the EEG in the brain, in different areas of the brain. So I can measure the effect of the cortical stimulation and what is the effect on the EEG of the patient. So it is very, very good tool because we can see the effect on the motor, the effect on the EEG, central and peripheral effects of a molecule. Interestingly, for example, you can see here in this slide, it has been done, this resting motor, that is what I show you here, the motor. This is the motor and this is the EEG. This resting motor was studied, for example, with reticabine. So a dose of reticabine caused this change in the resting motor in a patient. If you give, reticabine to a patient and you create this stimulus, there is a decrease in the resting motor. So you prove that you decrease the cortical excitability. The cortex is less excitable if you do this. Of course, there's always the question, okay, if this has something to do with the nerve, something to do with the external, it's really central. But yes, most probably it's really central. I will show you something that confirms that it's central. But we tested, in our studies, we tested xenon 1101, and we show that we have even significantly more effect in this resting motor threshold. Now, be careful, please. This is not the same study. Different studies, and scientifically, talking, it's not appropriate to show things like this. I'm not comparing one against the other, because this is not correct. But I'm just putting them one next to other just to show that this mechanism it works and has been proven to be, active in 1101. But when you do the same, as I show you, the EEG effect, I'm not asking you here to read the waves, but believe me, clear, significant effect on TMS EEG with 1101. And I will show you a little bit more detail on that. This is in our phase 1 study. This is what we did, how showing increasing doses of 1101 have an increased response in RMT. What is beautiful, showing how you increase the dose, you have better response. And when you see the EEG, this is the EEG response, and these are different waves that you measure in the EEG, in the TMS. You see here, it's not for us to clearly understand that, but just I wanted to show you this slide. You see here pre-dosing, you see here post-dosing, and if you compare the pre and the post-dose, it is clear, significantly statistical difference between active and placebo. So when you give placebo, you don't see the same difference that when you give 1101. So clearly, there is an effect in resting motor threshold, meaning the motor activity, and is an effect on EEG, what clearly shows that the effect is central, decreasing the cortical excitability. So this shows that this molecule really has the opportunity to go into the brain and have an effect on the cortical excitability. Now, what do we need to do? Drug is there, the mechanism of action is clear, we are having a clear effect in the EEG, a clear effect in the resting motor threshold. We need now to prove this drug works. We need now to prove this drug can bring benefit in patients. So we need now to go and see if this is really an effect that a drug can help patients in epilepsy in general, but also we need to see what is the safety profile of this drug. What happens if this drug is going to cause the same urinary retention? We don't think so. We have some explanation why that, but we have to explore. We have to study. We need to develop the formulation. Of course, we are working in that, and we are going to initiate our phase 2 study in adults with focal epilepsy. And I think this is a huge step. We're going to be studying our phase 2 study in focal epilepsy before the end of this year. And that's our commitment, working very hard to make that happen. So we will have very soon data on efficacy in epilepsy and safety profile that can guide us a lot in our next steps. And of course, we will start exploring other indications after that and being, what is the possibility to bring then this opportunity to KCNQ2 and other epilepsy or encephalopathies. So we are very happy and committed that we are coming back with this another molecule that has an improved profile comparing with ezogabine. So in summary, ezogabine is a drug that has been proved to be efficacious in epilepsy in the past. We are working very hard in a pediatric formulation. Our plans to do a phase 3, go directly to phase 3 study in pediatric population, really serious. We are working towards that objective. We need to work, as I said, and I want to repeat, in the pediatric formulation to be sure that we can overcome that difficulty. And this is definitely a team effort. We need everybody. It's not easy to do a study. This has never been done. And I think that's a very important thing. This has never been done. A study like this, placebo-controlled trial, to prove an indication in a disease like this, with this type of design in epilepsy, has not been done. So it's difficult. It's complicated. But we are committed to do it. But we need you, we need the foundation, we need the experts, we need everybody to help us to be sure that we can explore this opportunity and bring it back to reality and help patients. And our Xenon 1101, of course, is moving fast. We are going to start our phase 2, as I said, soon. And I think it's a drug that is coming back and it's coming behind, and we may have an opportunity to help also in this complicated scenario. I cannot say thank you to everybody. I mean, I will forget someone, but the foundation, I really want to thank you. It's an incredible, every time that we on and we talk to the foundation, it's amazing. The foundation motivates us every day. The foundation, every time that we talk to any person in the foundation, we feel more and more inspired and more committed. I think that you're contagious. You just really, give us the energy to continue and move forward. So thank you very much, the foundation, for everything